Okay, before we talk about translation, there's a couple smaller topics we need to talk about first, and one of them are codons. One of them is the topic of codons. So first, take a moment, stop the video, and talk yourself through the processes of transcription and messenger RNA formation using these pictures, which you've never seen before. There's some words there to help you, and there's some terms for you to mention. The reason why I want you to do this is because you need to be able to talk yourself through it in order to take a test. And remember, we're getting really close to test time now. All right, thanks for doing that. So now let's get back to codons. So now when we're looking at messenger RNA, and we know we have all those nucleotides, 3 consecutive nucleotides on the messenger RNA constitutes a codon. So there's 3 right next to each other, that is a codon. And what a codon is going to do, it's going to specify a particular amino acid. Now, remember, this is only when we are talking about messenger RNA. Has nothing to do with DNA. It's going to have nothing to do with transfer RNA. A codon is 3 consecutive nucleotides on messenger RNA. So if we take G, C, A, and U, the 4 nucleotides that you can find in messenger RNA, and we put them in every possible combination, there are 64 possible codons. And yet there's only 20 amino acids. So what does this mean? What this means is most of our amino acids can be coded for by more than one codon, meaning there's duplicity built in. So it's kind of a good system. Out of these 64 different codons, only one is a start codon. So in other words, when you were reading these codons, none of the codons reading them, because remember, we start at the 5' and we go towards the 3'. When you read the codons, you get to ignore them until you get to the start codon. And the start codon is AUG. So anything before AUG is like, well, that's nice, that's nice, that's nice. It's like, oh, now I need to pay attention. Okay. And the start codon is where translation will start. No translation is going to occur before that. So no protein chain is going to begin until you get to the start codon. And AUG codes for methionine. So what does this mean? No proteins are coded for before AUG. And since AUG codes for methionine, every protein starts with the amino acid methionine. Pretty cool. And then the next codon will code for a protein. And this is going to keep on going until you get to a stop codon. And there are 3 codons that don't code for amino acids. They are stop codons, UAA, UAG, and UGA. Now, you don't have to memorize what the start codon is or what the 3 stop codons are. Aren't you happy to hear that? Because they are written for you in the same table that tells you what the codon is, what proton is for the codon. The same table tells you, oh, this is the start codon, and oh, this is a stop codon. Okay. So for instance, in this little strip of messenger RNA, here's our start codon. So there's methionine. AAG would code for lysine. UUU would code for phenylalanine. And UAG is a stop codon. So this protein is only 3 amino acids long. Okay. If there are codons after this, and one would assume there would be more codons, any codons in a messenger RNA after a stop codon will not add amino acids to the protein because the messenger RNA is going to disconnect. So how do we decode the codons? We look at a codon table. So when we look at a codon table, you will notice there's C, G, A's, and U's written on all three sides. So we start on the left side where we look up the first space, and then we go to the top, and then we go to the third. And so if it's a G, U, so it'd be G, and then U, and then the third one is there. And so if we're starting with A and then U and G, you can see AUG here in green, methionine, and in parentheses, it says the start codon. Okay. And then here in red, you can see stop codons for these two, and there's the third stop codon. So you can see the 64 different options for codon, and yet there's only 20 amino acids. Methionine is only coded for by AUG. Tryptophan is only coded for by UGG. And yet leucine, you'd go, oh, that's coded for by 4. In fact, leucine is coded for by 6 different combinations. Serine has 4, proline has 4, threonine has 4, valine has 4, alanine has 4, glycine has 4, and quite a few of them have 2. So there's a lot of duplicity here. Now, I want you to use this table, and here is messenger RNA that went with this DNA. So here are the codons. I already given you the codons, and what I want you to do is decode these codons and then figure out what the amino acids would be. When you're ready, yes, then you can look and double check your answers. You would have 7 amino acids. Well, actually, you will only have 6 amino acids because the last one is a stop codon. Okay. Now, in addition to the codon chart, there is something that's called a codon wheel, where to read the codon wheel, you start in the center with the first, and then you go out, and then you go further out. Okay, so I want you to practice using the codon wheel. This time, I'm not giving you the RNA codons, I'm giving you the DNA. And unlike the last one where you had the answers written on your sheet of paper and on the slide, this time you don't have the answers, so you're going to have to work through them yourself. If you have any questions, write to me through the Canvas inbox. What would be your RNA codons, and what would be the protein that is coded? Don't forget, you don't code for a protein until you have a start codon, and you stop with a stop codon. All right. And when you're done, we are done with this section. So thank you for doing the work involved in this course, and I will see you shortly as we continue on with these concepts in this section of human biology.